Now time for the next speaker. It is Dr. Niall Lennon. Dr. Niall J. Lennon is the Senior Director of the Genomics Platform and an Institute Scientist at the Broad Institute of MIT and Harvard, and CSO and Chair of the Board of Managers for Broad's Clinical Testing Laboratory, Broad Clinical Labs. Dr. Lennon is also an Associate Director of Gairdner Center for Cancer Diagnostics. Since starting at the Broad in 2005, he has contributed to the development of applications for every major massively parallel sequencing platform across a range of fields, including developing the Broad's own clinical diagnostic COVID-19 testing process that delivered 37 million diagnostic test results to people in the New England region. Please join me in welcoming Dr. Niall Lennon.
Hi, everyone. Thank you so much for inviting me to come here today. It's really an honor. I'm going to set a little timer for myself because I tend to get carried away and excited. So I'm really delighted to be here. I'm going to tell you a little bit about the National Biobank program in the United States called the All of Us Research Program. But before I get to that, I'm going to give you maybe my version of the context of genome sequencing and the technologies that have brought us to where we are today. And I should mention, I can speak about the All of Us Research Program. I'm not speaking on behalf of the All of Us Research Program. I am one of the principal investigators of one of the awardees of the All of Us Research Program from the National Institutes of Health in the United States. And we are a genome center. We do genome sequencing for the program. But I'll tell you a little bit about what it does. So just to say what I do, first of all, so the Broad Institute of MIT and Harvard is a nonprofit institution that was formed just 20 years ago. And in fact, we had a little party last night to celebrate 20 years of the Broad Institute. And it was really built on, we were fortunate to be part of the international consortium that did the Human Genome Project. And based on that, with a philanthropic gift from the Broad family, Broad is a family name, the Broad Institute was born to accelerate the use of genomics and biomedical research in general. And it was formed as part of both MIT and Harvard and is currently an independent nonprofit research institution. About 10 years ago, I formed, with leadership at the Broad Institute, a clinical laboratory within the Broad Institute, which is now called Broad Clinical Labs. And we reflect the mission of the Institute as a whole, but we also work in that translational space. And like Christian, we can process samples and return results to patients as well. But really at heart, I would say, I'm sort of a technologist. I really love the technology that has contributed to everything we can do. And really, we see our role as connecting the scientists who really have questions about biology and disease and human development and function with the technologies and the data that they need to answer those questions. And the types of scientists and researchers we work with span a few different areas, what I would call sort of genomic explorers. And these are people who do some of the work that Christian talked about and Eric, in that it's really looking at large data sets of people with certain diseases and people without diseases and looking for those, discovering those mutations or genetic causes that might be underpinning those human diseases. And that can span common diseases and also rare diseases. We work with groups who are doing what we would call resource building. And so the All of Us program, a national biobank program, is largely seen as a resource. It is a resource for the community to use. So data sharing, the idea of being able to access large amounts of data from well-characterized samples, whether that's genome data, RNA data, other types of data. Those are resources that institutes like ours help to build, largely with funding from government sources. And then a lot of what we would say is clinical translators. So, I've been working in this field at the Broad Institute for 19 years. When we first started, all of the data generation we were doing with genomic technologies was really for research. But over the years, as more started to be discovered and more was known about the implications of what happens when you have a certain mutation in your genome, it became more clear that we should be telling people about that. The way to do that is you have to generate that data in a clinically regulated environment. And so that's what the formation of a clinical lab like Broad Clinical Labs allows you to do. And so we have a lot now of activities where discoveries, new molecular assays get translated into something that can be used for the benefit of patients. And that can go through a process of, it may not be directly to do diagnostics on day one, but it may start off as being clinical research, clinical trials, then you go into sort of clinical utility studies to show that it's actually of utility to do this analysis, and then returning, ultimately it may make its way into a diagnostic assay, or in some cases an actual therapeutic assay. But like I said, I'm sort of a technologist at heart, and I really like to nerd out about the actual technologies, and it's really an honor to be able to speak to an audience that includes the laureate from this week, who were sort of pioneers in developing the chemistries that we use today at really large scale in the instrumentation we use to sequence genomes. We are one of the genome centers, like Christian described, where we essentially, you could think of us as a genome factory. We're essentially a building with a lot of really expensive but really cool machines in there. And I often say that we convert the biological into the digital. And that means that what comes in on our loading dock are blood samples and tubes with saliva in them, and pieces of paper with blood spots and stool samples and all kinds of biological squishy things that come in one end. And what comes out is, in fiber optic cables, it's all information. And what we do is what's in the middle. And how we do it is using the technologies that have come, that are now largely known as next generation sequencing technologies. As an aside, of course, people have tried to get away from the term next generation because we kind of painted ourselves into a corner there. And you'll often hear the term massively parallel sequencing technologies, but NGS, or next generation, has kind of stuck. So we've been in the next generation for probably more than a generation at this point. But regardless to say, now, for a long time, the short read, what we've been calling short read technologies, which really came from some of the inventions that led to Solexa and ultimately a company called Illumina, were sort of the largest players in the field, and those were the machines that most people use. And in the clinical applications that Christian talked about, and in our clinical lab, it is those short read Illumina technologies that are predominant still today. But there are other types of sequencers now as well, other types of instrumentation from other companies, and they have different attributes. They can have, again, you can hear the term short reads. A read is just the unit of data that comes out of the machine, right? If we think of the human genome as a giant jigsaw puzzle, the read is that little piece of A's, T's, C's, and G's that comes out of the machine. And the read, if it's a short read, that's one thing. If it's a longer read, it's like a bigger jigsaw puzzle piece. It's a little bit easier to analyze, a little bit easier to put together. But they all have different attributes. And as Christian pointed out, the bigger the read, the longer the chunk, the more information, but it's also currently more expensive. And so researchers have done a lot of development to be able to do a lot with the shorter reads. All right. So before I get into the All of Us program, I do want to sort of highlight a little bit about how we think about human whole genome sequencing. And there's lots of other types of sequencing. There's environmental and forensics and infectious disease and microbial, lots of other things. But I think today I really want to just highlight how sequencing the human genome has impact across the whole span of our lifetimes. And you can use this same piece of data, essentially, and these same pieces of highly engineered instrumentation to discover lots of different things about all parts of our existence. Starting from the earliest sort of embryogenesis, where embryos are developing in the womb, we often think of ourselves, and we sort of teach that the human genome is essentially the blueprint of life, and that every cell in your body carries the same genome. It's not quite true. From the very earliest days, different cells, as they start to divide and develop into organs, will actually start to accumulate very small amounts of differences. And this is known as mosaicism. So essentially, we're not kind of a single entity where every single cell is exactly the same. We're actually a mosaic as we walk around. And if you want to get into microbiome, we're really a mosaic because there's all kinds of other stuff that walks around with us. But just within our own cells and our own DNA, you actually can have slightly different sequences in different parts of your body. And as we get older, those differences accumulate. And it differs by which organ you look at. And if you think of some obvious ones, so skin is our largest organ, skin accumulates a lot of different mutations over life because it's exposed to radiation on a daily basis. Whereas some other organs that are hidden away accumulate less. But there's lots of, now, by sequencing the genome of these different tissues, you can start to look at these patterns and signatures of mosaicism within our body and understand how these relate to normal aging, but also disease processes. And so that's a really exciting kind of maybe newer area that a lot of researchers are starting to move into. We've heard a lot about newborn screening or newborn sequencing already, and it is a really interesting area that I do think we'll hear a lot more about in the coming years. The idea that the cost of genome sequencing has come down to a point where you could sequence, if you do it at scale at a national level, you could sequence the genome of newborns for about the same cost as all of the other standard testing that is already done on newborns. So most newborns, certainly in the United States, get a heel prick and a drop of blood put on a piece of paper that goes off for biochemical and enzymatic testing. And human genome sequencing has gotten to the place where it's almost about the same price of doing the genome as the other tests. And the big question, of course, is, number one, are you faithfully replacing all of the existing tests because they're existing because they're very effective and they're cheap in their own right? But you in theory, would learn a lot more by sequencing the genome at birth. And the things you might learn that are acutely important are the things that are important to know in a newborn, things that you need to take action on immediately to avoid certain things like hearing loss and other things. But there's also things that then you could come back and look at later that would be important later in life. And then you have this concept of you only have to sequence sort of once for the most part. And you could look at that data many times over your lifetime. And that's an interesting idea. It's more likely to be implemented in countries that have a centralized single-payer health record system, which is not the United States. But you, of course, have to overcome all of the concerns, legitimate concerns about things like privacy and sort of genetic destiny and determinism and other things that have to be taken into consideration. But there are many projects that are looking at newborn screening. And of course, in the acute cases of sick babies, you've already heard from Christian, and there are still hospital systems now who are routinely doing sequencing of genomes in their NICU units for very sick babies who have something that might have a genetic cause, and the idea of doing it very quickly and learning things could be really important. Rare childhood diseases is a big area for genome sequencing. The idea that instead of doing what used to be the standard of looking in one gene because there's a certain phenotype that somebody has described in the literature matches that gene, and then that's not the case, then you look in another gene, this so-called diagnostic odyssey that can stretch on for years. By sequencing the genome, you can sort of shortcut that journey and give families some information. The idea of replacing existing tests, like we do a test called chromosomal microarrays to look for certain types of developmental delay. You could likely also look at that in the genome. I'll talk about the idea of sequencing populations to look at population health and also to really capture, to understand the associations of genetics and disease in diverse populations that actually reflects the ancestral genetic diversity of the human population. There's lots being done right now in looking at our genome to understand our risk of disease. And so, many chronic diseases now you can look at. There are well publicized cases of looking at a certain gene, like a BRCA mutation, and your likelihood of developing breast cancer. But there are also these other types of risk known as polygenic risk, where it's not one gene and one mutation, but many different signals throughout your genome that could significantly increase your risk of developing certain common diseases like cardiovascular disease, prostate cancer, breast cancer, diabetes, and that if you could tell somebody that they were in that high risk category before they developed that disease, they could start to monitor, do more frequent screenings, change their lifestyle, that we could actually save the healthcare system money by investing in preventative medicine instead of treating the symptoms. Of course, cancer is a disease of the genome, and there is a lot of application in sequencing of genomes for cancer, both in sort of diagnostics and therapeutics and personalized therapeutic regimens, and of course in lots of common diseases, and some of the maybe more exciting things that have happened over the last couple of years based on large population and case control studies are starting to chip away at the underpinnings of psychiatric diseases, schizophrenia, bipolar disorder, which have been sort of harder to crack over the years traditionally. So across the board, there's like so many different applications of this just one assay using this one set of technologies that it's really amazing. And at our own institution, in our own group, we have projects and partnerships in all of these areas with groups all around the United States. All right, so switching to one of those such programs, the All of Us research program in the United States. Like I said, I'm a awardee of a genome center, so we do data generation for this program. The program is an NIH program. It actually has its roots back in the Obama administration. Some of us might like to call it the good old days, where it was called the Precision Medicine Initiative. But it was really this idea of gathering a large cohort of information from the US population to help drive research into diseases that affect that population. And it was not fully modeled on, but certainly took influence from prior programs like the UK Biobank, where the UK has had programs like this, which started earlier and have already been used to great effect. But one of the things about sequencing populations is that you kind of need to sequence many different populations because the UK population doesn't fully reflect the United States population. It doesn't reflect the Canadian population, the South American population necessarily. Those countries sort of have their own mix of genetic ancestries. So information or associations you might make by looking in the genetic data of the UK Biobank are really powerful predictive measures for people who have genetics like people who participated in the UK Biobank program. So for me, I grew up in Ireland, I'm Irish, so Ireland is not part of the UK, to be very clear, but genetically very similar. And so an inference made from a UK Biobank data set is great for me, but if you're Southeast Asian or from South America or another population, it may not be as accurate. And so one of the things that a lot of people who work in genomics really care about is the equitable sort of results, that things we discover and things we build should be equitably beneficial for everybody. So one of the mission of the NIH All of Us program was to really capture the diversity of the US population so that discoveries and associations made are accurate for the population. And it's a nuance, but just it's important to point out, I guess, is that when you look at genetic ancestry, other cohorts and groups of data have been put together to be diverse, quote unquote diverse, but in some ways that meant we took a bunch of people from Europe, and then we took some people from China, and we took some people from Nigeria, and we took some people from India, and now we have a diverse data set. But actually, if you look at the diversity, certainly in the United States, it's not a pocket of people who are 100% one thing. We have this what's called admixture, is that my grandfather is from China, but my mother is from Europe, or my grandfather is from the West Indies, and my mother's from Australia. And what does that mean, right? That admixture, that's just human nature. We're not sort of, my family's like 100% Irish, like they never left anywhere, but many people have lots of contributions to their genetic ancestry. And so studying populations is really important. Okay, where's the program at? So it did a big push on enrollment, so it's enrolled a lot of people. The goal was to get to 1 million. They've at least enrolled that initial contact, we're getting close to that, and then about half or 60% of those folks have gone on to do the online steps of consenting and saying they'll submit their samples. One of the things you consent is to link your electronic health record. That's really important for these kinds of studies. It's great to have lots of genomes, but if you don't know something about the person's who gave you their genome, what diseases they have, and other things about them, it's less useful. And so you really want to capture as much information as you can about the folks who have signed up for the program. So when you do sign up, you do things like you consent that your electronic health record can be sort of sucked into the program, and that measures that are in your medical record can be associated with your genome. You can answer surveys about your lifestyle, where you live. There's a big focus on social determinants of health. There's a lot of studies around sort of, parts of the country where you live, where you have less access to healthcare, and what's the impact of that on your health. And there's a lot of interesting study, if we talked about epigenomics, about trauma and stress and what impact that can imprint on your genome and how that gets passed on to different generations. And so you need to capture all of that information to make these discoveries. And then physical measurements, you provide biospecimens. In some cases, you can link your wearables, and it can monitor your activity and sleep cycles and things like that. And so in terms of data, so the important thing for the research community is that this data is accessible, but even for the public, you can actually go and browse the data. So there is a data browser for the All of Us program. If you Google All of Us data browser, you'll find it. And anybody can just look at sort of aggregate sort of metrics and how many people are there in the cohort with breast cancer? How many people are there who've said they have atrial fibrillation? And you can just see the numbers, and you can say, well, what is the age group of those people? And you can sort of slice and dice the data a little bit. And that's a way of looking in there. Researchers, so people who come from institutions that sign data use agreements, it's not a cost, but it just says you'll use the data ethically. You agree to many sort of protocols about what you will and will not do with the data. You do a lot of training, online training about ethical use of the information. And then so researchers who qualify for that can register to access some of the other data. So the electronic health record data, not with people's names and addresses, but the fields. And then also in an even more controlled tier is the actual genomic data. So people can access that as well. And to dig into that tier, so that All of Us data set does now have one of the larger data sets of human whole genome sequences widely available for research. Again, the UK Biobank is another great resource that's been used widely for this as well. But there are, in the latest build, about 250,000 whole genome sequences. That will go up close to 500,000 soon. And then those are associated with lots of other pieces of information. There is also some data on long-read sequences that we've added to that. We've also actually added, it's not in the data set that's available yet, but we are adding things like proteomic data, so other omics, other things that are in your body, circulating in your blood, and RNA sequencing data as well. So you can actually really start to explore the omics for discovery. So that's what's available to researchers. And it's impossible to go through what everybody is doing with it, but you can also look at, there's a research projects directory where researchers who go through the program to get access to the data, write in what they're doing, what their research study is, and there's over 13,000 active research projects happening in the data. One of the early criticisms of the program, and it wasn't a malicious thing, it was just a logistical thing, was it was only initially available to researchers within the US. In the last year, now it's available internationally, so any accredited sort of research institution in the world can apply for access to the data for their researchers. And also now actually companies can start to apply for access to the data, because pharma companies might also want to start looking in this data to find people with interesting attributes. And UK Biobank has done this very well in actually encouraging industry to fund some of the data generation to help populate clinical trials and advance therapeutic development. And so lots of really good projects happening. I'll only highlight maybe one with the time I have left, which is one that I did, self-serving of course, but that's the one I know most about, and actually spans some of what we've talked about already. So sort of a busy slide, but just to talk about a study out of Eric's institution, the national NHGRI, which is called the eMERGE network, which is Electronic Medical Records and Genomics, which is a distributed network of 10 academic medical clinical centers around the United States who enroll patients. And what we do in this study is we've been looking at sort of the utility and what do people do if you tell them they're at risk of developing certain diseases. So this is this polygenic risk that I talked about earlier. And so there's 10 conditions: asthma, atrial fibrillation, cardiovascular disease, diabetes, prostate cancer, breast cancer, and so on, that get returned, obesity, that people who sign up and consent for the study, we actually genotype their sample, and then can apply these models from these large case control cohorts to understand whether they are actually at an extreme risk, sort of the tails of the distribution of risk, at an extra high risk of developing one of these diseases, even if they don't currently have it. And so that's a really powerful thing, and actually part of the whole study is, number one, do people understand that? Number two, what do they do with the information? Do they actually get more frequent screenings? Do they change their lifestyle? Do they do something about it? So that's sort of outcomes research. But also, do the doctors and nurses and providers understand what the test is also? You know, I'm in Boston. My primary care doctor is at Mass General Hospital. Mass General Hospital developed a cardiovascular polygenic risk score and published it. And my family has a history of heart attacks. I'm like, well, I want to know. You know, I'm 48 still. My father had a heart attack at 46. I want to know, am I at higher risk of, I mean, I know I'm at some risk because of my father, but genetically, am I at higher risk? I went to my primary care doctor, and he's like, a what? A polygenic what? He had no idea, even though it's developed at his hospital. So it takes a long time to educate the actual people providing healthcare in the community about the possibility of new types of tests, and that's exactly what this study is really about. But one of the things we talked about is many of the scores, these actual sort of algorithmic models that tell you if you're at high risk or not high risk, are developed in certain populations. And they're more accurate for those populations that they're developed in. Some of them would be developed in the UK Biobank data sets, some in other data sets. And so this idea of transferability across genetic ancestries is a big problem. And so what would happen is somebody who came in who's from a different genetic ancestry than the score was developed in might end up on somewhere on this sort of risk distribution curve that's not at the tail end if you compare them to everybody else. But if you compare them to everybody else who looks like them, they would actually be at the high end of the risk. And so it's important for them to know they are actually at higher risk. And so it's hard to do that unless you have giant data sets to model the score in every possible genetic ancestry, and again, with admixture, which is just a continuum. And so what we actually were able to do is take all of those scores, bring them into the All of Us data set, which has a lot of admixture and a lot of people, and then build the models, the mathematical models to essentially calibrate the scores so that people, regardless of what genetic ancestry they came from, if they're in that high-end tail of the distribution of risk, will find out about it. And so that's one just very simple use case of why having a large, diverse biobank data set is really useful as you get to trying to implement new things in clinical genomics. Okay, I think I will end there. I just want to make sure I thank everything we do. You know, I feel like we have the best jobs in the world here. We're really connecting really cool technologies which are developed in these multidisciplinary teams from engineers and mathematicians and chemists and biochemists and everybody else, with scientists who have burning research questions, with society and patients and people who have real world problems who want to contribute their samples to help find insights and ultimately cures for those conditions for society as a whole. So thank you all for your time.
Thank you very much for the presentation and our introduction to the All of Us program. So first question, when we sample tissues for clinical sequencing, should we be concerned that we are missing important variants due to mosaicism, i.e. variants not captured in the specific sample?
Yeah, I think it's probably a little bit too early to worry about that just yet. I think most of the things we know, the associations between, you know, a single mutation or even a polygenic score from a blood sample, which represents many, many, many genomes or copies of your genome, will be representative enough. I think as those studies into somatic mosaicism advance, it'll start to get just a finer resolution, and that'll get into things like single cell sequencing and other. It's just, you know, finer and finer resolution of information that in the future might start to become important. But today, a blood sample or a tissue sample is generally representative enough to give you the answer you're looking for.
Perfect. And I think one of the questions is, what is the major challenge in recruiting 1 million participants?
Yes, well, I mean, there's lots of challenges, money being one of them. Actually, the All of Us program did a tremendous job early on of engaging community groups. And so one of the things I mentioned but didn't go into detail was at the start, the All of Us program really wanted to fill the cohort with people who were underrepresented in biomedical research. If you live in the Boston area, and it might be similar to this area here, and you go on the subway, you'll see ads all the time. Join our research study. Come and, you know, join research. So you get asked all the time if you live close to a big hospital. But if you live in the middle of, you know, a rural area, you may never get asked to be part of research. And so all of us really want to try and get that wide net, and certainly in areas sort of maybe in the southern US with more diversity. So they had things like a bus that would drive around, they would have events at community clinics and other things. And that's how they really got the word out. And then, advertising on a website, allowing people to sign up online, sending them spit kits to their home so they could mail it back, making it as easy as possible. Okay,
perfect, thank you. And how can we use the eMERGE network to better educate the next generation of healthcare professionals?
Yeah, so now we've actually just about finished all of the data generation part, so the testing part. So the eMERGE network enrolled about 25,000 individuals across those 10 clinical sites. Our lab we've delivered 155,000 individual PRS test results to participants. But now all of those clinical sites are returning those to the patients, and they'll do these things of monitoring and surveys and asking questions and saying, what am I doing with the information? Is this outcomes research? And all of that will get published, and the papers will start to appear and they already have, of what is the perception, what do people do with the information? That'll all get published, basically.
Okay, perfect. And what collaborations do you have regarding biomarker discovery?
Yeah, it's a great question. So many of our researchers are doing biomarker discovery, and that's really when you have these case-controlled studies, right? You have a lot of people with a certain condition, many people without it, and you start to look at what are those signals, and it could be a mutation, it could be a signature, it could be a proteomic thing that's associated with that disease. And so some of those common disease breakthroughs, like we said, in like the psychiatric disease, have really been those sort of biomarker discoveries of psychiatric diseases. And so we work with the researchers who are doing that in a research setting. We also work with pharmaceutical companies who are doing that in their clinical development programs.
Perfect, thank you. And we do have one or two more questions we have time for. One is all of us is looking at a lot of parameters, wearables and other parts, and I think you hinted briefly about microbiome. So do you see a future where you can sequence both genetics and also microbiome to kind of get a better understanding of an individual?
Absolutely. The All of Us program itself doesn't collect the specimen for a microbiome profile, but we actually do have a big program on microbiome profiling in our metagenomics program. And so that when I say we get stool samples, like frozen stool samples are one of the things we commonly get in. There's a lot of studies in gut microbiome and how the populations of microbes that live in our bodies impact our health, and how dysbiosis of those populations may lead to chronic conditions like inflammatory bowel disease, Crohn's disease, C. diff infections, and things like that. So it's a pretty active area.
And last question, is there a value in being able to collaborate between Canada and the US in creating richer biobanks?
Absolutely, yeah, of course. I think there's value sort of internationally in everybody sharing their data. It's the same issues that Christian brought up. It's just where does the data live and how do you access it? The All of Us program has spent a lot of time in their infrastructure allowing people from around the world are able to access the data now. And so, in an ideal world, you would be able to join your sort of, Canadian Biobank and the All of Us Biobank data together. There are technical issues around that, but you can certainly project the findings from one into the other and things like that. But there are many people who spend a lot of their time really building collaborations and networks for genomic data sharing.
Thank you very much.
Nope.